Hello to everybody. It's 10 a.m. Houston time, so it's Enjoy Science time. Thank you, all of you from Houston, from United States, and from all over the world who connected, because without you, Enjoy Science make no sense. So this is a webinar designed by scientists and clinicians from MD Anderson for all large family of friends, patient, clinician, scientists who know us and who work with us all over the world, also inside MD Anderson. And it's hosted by me and my colleague and friend, Dr. Alessandra Ferraioli. I have to thanks to Dr. Giulio Draeta, who organized this webinar, and also to Alison Garth and Liz Kent, because without their very hard work, nothing can be done. Also, I want to tell you to the panelists 2 points. First, during the presentation, please use the chat system to ask questions, because without questions, the presentation makes no sense. And second, this presentation is recorded, so you have the chance to go on the webpage and see it well. And with this said, it's my great pleasure to introduce you Dr. Jim Ellison, and it's a very simple task. Why? Because Dr. Ellison is from the handful of scientists in the history who you have to say only the name. Everybody will know what he has done for science. Of course, we are thinking to CTLA-4. We are thinking to checkpoint inhibitors. We are thinking to the Nobel Prize. But let me to tell you personally what I think is his big achievement. A few weeks ago, in the most high impact factor journal, CA, a journal for clinicians, it was showed for the first time in the history that a human cancer, in this case metastatic melanoma, mortality dropped significantly starting with 2013. And this is exactly because of the discovery and the use of checkpoint inhibitor for patients. So I can say you without being wrong that the work and the science of Dr. Ellison and his colleagues, it's saving millions of lives now and many, many other millions of lives in the future. And I would say this is the biggest achievement of Professor Ellison, and for this one, he is respectful and really we are loving him unconditionally. If you want to learn more about his driving, about his belief in his discoveries when nobody was believing, please go to PBS this month of May. It's a free documentary named simply Jim Ellison Breakthrough, showing all this wonderful journey to saving millions of lives. With this said, Dr. Ellison, thank you for your time. Thank you for being with us, and please run the show.
Okay, well, thank you, George. I want to also Liz and Allison, who helped me master technical difficulties of getting this going. And I think this Enjoy Science thing has been a wonderful thing during our period of the COVID-19 isolation that we've all been undergoing to kind of keep us together. But it's been so good. I hope it's something that we can keep going during the year. So anyway, with that, my disclosures are here. What I want to do today is provide first a little bit of background on where checkpoint blockade came from, a little bit of kind of where we are now. But most of it's going to be spent on talking about really mechanistic, cellular, and a little bit of mechanistic details about checkpoint blockade in general, but also specifically the large number of differences we find between the effects of CTLA-4 and PD-1. And I hope when I finish this, you'll be convinced that these are really two separate drugs that need to be considered that way. And particularly because I think we've got some insight on understanding why they work so well together. So I'm going to start with acknowledgments first. My lab members are shown here, but most of the work I'm going to show today is done by former postdoc of the lab, Spencer Way. And at the last, I'll show a little bit of work by a current postdoc, Stephen Monk. And other collaborators are shown there, and also a lot of this work has been done in collaboration with Pam Sharma, and with some help with the computational stuff from Donna Pear, who's now at MSKCC, and the funding's on the right. Most of this has been early work by the sources up there, but current work was funded by CPRIT and the Parker Institute. And here's my lab. The guy sitting next to this handsome guy here is Spencer, and there's Stephen there in the rest of the lab. So hi guys, I haven't seen you in a while, but anyway, we'll get back together in a week, hopefully. So this work really started at basic science. It was not anything to do about cancer at all. It was trying to understand the basic biology of T cells. And one of the things that we had learned after identification of the T cell antigen receptor was that that signal isn't sufficient to activate T cells. You actually have to have that, plus co-stimulatory signals that we showed in the late 80s through work in the lab at that time by Jane Gross and others, that was supported by co-stimulatory mediated by CD28 on the T cell surface, recognizing B7 molecules shown by a number of other people on antigen presenting cells, especially dendritic cells. But by the early 90s, we had shown that one of the reasons that tumor cells are so hard to attack isn't because of lack of antigens, which is what people had thought for a long time, but it was because of lack of ability to co-stimulate. And we showed that, Sarah Townsend in the lab, by putting B7 molecules into tumors. And we could show that tumors, which otherwise would grow and, and kill mice in a matter of weeks, if we put B7 into them, they wouldn't grow at all, or they would grow for a very brief period and then be eliminated. And so again, the lack of responses weren't due to lack of antigens, it was due to lack of co-stimulation. And so that just, on this cartoon, that just shows that, the tumors are invisible until they die, and probably by necrotic death normally. Eventually, they get too big and don't get enough energy, food supply, oxygen, whatever. They die. That causes inflammation, which attracts antigen-presenting cells, largely dendritic cells. And then a process called cross-priming goes on. Since the T cells can't recognize the tumor directly, what happens is the dendritic cells process antigen bits and put them on the surface in the context of B7, and that leads to signal 1 and signal 2, which induces a whole program of genes that lead to IL-2 production and other things, and proliferation, expansion, differentiation, which I'll talk about a little bit later. And that's been known for a while, but by the mid-90s, we also learned from a work of Max Crummell in my lab and also Jeff Bluestone's lab. There's a close homolog of CD28 called CTLA4. That's an inhibitory molecule on T cell, and its expression is also induced by signal 1 plus signal 2 of CD28. So you turn on this off signal, and with time that accumulates to a level where CTLA4, we believe, outcompetes, because it's got about a 3 or 4 log increase advantage in avidity for the B7 molecules, and that stops co-stimulation and can attenuate or eventually terminate T cell responses. And so we thought that might be it, because it's a race. The tumor's got a head start, and because of this clock that's built in with CTLA4, it's hardwired into the immune response. If it shuts the response off before the T cells have had a chance to eliminate the tumor cells, then the tumor wins. And so I had a simple idea: if we just block CTLA4 by putting in an antibody, we could pull that out of the program and let the first 2 signals dominate and have unrestrained proliferation for as long as we want. We could keep the immune response going for as long as we want. And when I had that idea, I thought it was really pretty compelling for a couple of ideas. One is, you notice we're not treating the tumor cells at all. This has nothing to do with the tumor cell. This involves targeting molecules on the immune system. That being the case, then the tumor is irrelevant. So this has the possibility of working against any kind of cancer. But, theoretically that's possible. In practice, we know, and I'll talk about later, that's not the way it works out. But it's a compelling notion nonetheless. And the other one is that because this has started with cell death and cross-priming, you could use chemotherapy, radiation, hormone therapy, angiogenesis, whatever you want to use in terms of conventional therapies. But while used by themselves, as soon as the drug's gone or you stop the treatment, if you didn't kill every tumor cell, it doesn't work anymore. In this case, we can use those to induce T cells, which not only expands the valency of the response, if you will, because you could bring into it T cells that attack, all the well, at least some of the antigens that are produced by mutations that are associated inevitably with the cancer process itself, and give them conventional therapies memory, give them this polyvalency that immunotherapy has, and so the possibility for combinations are pretty much endless. And so what we've learned since then is, unlike the first three pillars of cancer therapy, surgery, radiation, and chemotherapy, immunotherapy can work with all of them to give you a very powerful way of attacking cancer. But of course, the question is, ideas are a dime a dozen. Does it work? And I'm just going to show this one mouse experiment. This is one of the early experiments that Dana Leach did with Max Crummell in the lab with an antibody we made to CTLA-4, along with a C228 antibody that we'd made in the lab earlier. But in any event, what this shows is that tumors grow in mice. If you don't do anything, you euthanize the mouse after about a month. If you block CD28, the tumor grows even faster, showing there is a bit of a nascent immune response, but it just can't get the job done. But if we inject anti-CTLA-4 antibodies, the tumor grows for a while and then gets rejected. And the mice are permanently immune to re-challenge. When we got this result, I was just totally stunned. I thought we'd slow the tumor a little bit. But if you can imagine, out of all the things that are going on when the immune system detects a tumor, in the body, this one molecule, if you get it out of the picture, that's enough to get a full immune response and tumor rejection. Now, this tumor in this experiment was a tumor. It turns out, well, we knew it was pretty highly immunogenic. It turns out that it has a really high mutational burden, and there are other tumors that we used that are not so immunogenic that the monotherapy wasn't very effective against. But in those, we could add radiation, or we could add chemotherapy or a vaccine, and we never found really any kind of transplantable tumor of mice, C3H mice, strain A mice, whatever, BALB/c mice that we couldn't treat. And so this got us very excited. We spent a few years generating just a ton of data, and after a few frustrating years, engaged a company called Metarex that had a unique technology that they developed where they replaced the immunoglobulin genes of mice with human. So we could make an antibody of CTLA-4, clinic ready. So we did that, and this went into clinical trials. And in early clinical trials, it showed objective responses in many tumor types, as we hoped, including melanoma, and that's what I'll be talking about largely today, but also prostate, kidney cancer, ovarian cancer, lung cancer, etc. And then, of course, we had went to registration trial when Bristol Myers Squibb started collaborating with us, and then purchasing Metarex. But anyway, talk about that in a minute. But whereas in mice, though, we'd never seen any adverse events except a bit of colitis. A bit of vitiligo when we were working with melanoma, which we expected, but nothing else. And also in monkeys, the toxicity trials in monkeys didn't show any adverse events. But in humans, we know there are many adverse events: colitis, hepatitis, many pneumonitis, many itises. These are inflammatory reactions that typically, when the patients are treated with immunosuppressive drugs, and then they can be weaned off for symptomatic treatment, they can be weaned off of those and they don't come back. So these are not really autoimmune responses. None of these, except maybe hypophysitis in some cases, have ever been associated with a specific self-antigen target. However, but people with autoimmune disease were excluded from the trials. But now that hundreds of thousands of people have been treated, we are seeing examples of type 1 diabetes, whether this is from people that had subclinical disease. We don't know, but it's definitely typical type 1 T-cell mediated diabetes. And also more recently, a T-cell mediated myocarditis, which can often be fatal. We have recently developed a genetic model of that in mice, and I don't have time to talk about that today, but we've been using it, and I think we have pretty good candidates for treatment regimens for that. Work done with Javed at Vanderbilt, cardiologist. In any event, it was successful, and I'm not going to show you a lot of examples. I just want to show you this one because this is the first patient that I had met. She was 22 years old when she was diagnosed with metastatic melanoma. She had 2 subcutaneous tumors, 31 lung mets, and a brain met. And after a single round of treatment, you can see her cutaneous mets went away, but you can see here her lung mets went away, and also her brain met went away. She had a 0.5 centimeter brain met, and it also resolved. She had a complete response. I actually met her on the day that she was told that she was a complete responder, and that was quite a moving experience. But anyway, she went on, got engaged, got married, and had 2 children. And this is a picture from a few years ago. Her children are now teenagers, 2 lovely kids, and we're very good friends now. And I still keep up with her, and she's really doing fine. In any event, this went to a phase 3 clinical trial, which is shown here. This is a placebo arm here, 800 patient randomized placebo-controlled trial with overall survival as the endpoint. Progression-free survival couldn't be used because, as I showed you in the mouse experiments, the tumors often, and it was true in patients as well, tumors often progressed before they regressed. But nonetheless, this is a placebo control in this registration trial. It was not unethical at the time because there was no standard of care for melanoma, no metastatic melanoma, no drug had ever prolonged survival in a randomized trial. But the ipilimumab only arm, the top one, this dashed line, moved the survival over a few months, which would have been sufficient for our registration. But the cool thing is, this was a trial ran a little over 4 years. You can see that after about 2.5 years, the survival curve flattened out. Very, very intriguing observation, meaning very few people are dying after that point. Anyway, it was approved by the FDA in 2011, and then immediately went into a lot of trials. By 2015, there were enough patients, in this case, almost 5,000, with 10 years data from all the earlier trials, that there could be this follow-up analysis. And again, you can see about 3 years or so, the survival curve flattens out, stays at about 20% for 10 years. And actually, it goes on out. There's one patient from the phase 1 trial who was treated in 2001 who's still doing fine. So she's almost 20 years out now after a single treatment with anti-CTLA-4. And so I think we can use the word cure for describing people such as that, a word which at the time was pretty amazing to be used for melanoma, because when people were diagnosed with late stage melanoma, the median life expectancy was 7 months. And now about 20% of people living a decade or more. And that's exciting, but it's also disappointing. Why 20%? Why not everybody? Well, there's issues, there's mechanistic issues. Maybe there wasn't sufficient priming while the drug was on board. Maybe there were not enough mutations, not enough antigens to be seen, et cetera, et cetera. But of course, the most interesting idea was that maybe there are additional immune checkpoints. And turns out, as everybody knows, that is the case. There was a molecule programmed DATH1 was discovered by Tasuku Hanjo, cloned in about 1992 or something like that. And he thought that it played a role in negative selection in the thymus, but he ended a collaboration with Gordon Freeman and Arlene Sharp at the Dana-Farber, and they cloned 2 ligands for PD-1 and showed that it was another checkpoint, if you will. And so it's similar to CTLA-4. However, I want to point out that the biology of this, it turned out, it's been worked out by Li-Ping Chen. And one of the ligands for CTLA-4, for PD-1, PD-L1, can be induced on tumors by gamma, interferon gamma made by T cells. So T cells recognize an antigen on tumor cells, they make gamma interferon. The tumor responds by upregulating PD-1, which then functionally inactivates these effector T cells that they're inhibiting. Now, there's a lot of data suggesting that PD-1 plays a major role in protecting developing embryos against immune attack by the maternal immune system against paternally encoded antigens. And so it seems that tumors just adopted that method of acquired resistance to protect themselves. But in any event, we went into phase 1 trial, number of tumors, again, decent objective response rates in melanoma, in non-small cell lung cancer, in renal cancer, no responses in colorectal cancer, although Bert Vogelstein at Hopkins showed there was a subset of colorectal cancer that did respond. And the difference was that these had microsatellite instability, a lot more neoantigens, and they do respond. So that subset, colorectal cancer, does respond in larger trials, but also no responses in castrate-resistant prostate cancer. I don't have time to talk about that today, but that's something that that Pam Sharma's been studying with her colleagues in the immunotherapy platform at MD Anderson, and that mechanism for the lack of response has been worked out, and there's new combination trials that are underway that look pretty promising. But the reason I mention that is because that is a difference between one of the differences between CTLA-4 and PD-1 therapies, this lack of castrate resistance. Anyway, the fact that these are independent was underscored by the fact that patients who failed ipilimumab therapy and a CTLA-4 therapy and progressed could respond to anti-PD-1 therapy and vice versa. So this again suggests that there are different pathways. And of course, if you've got two different pathways, what do? You can combine them. Mike Curran in the immunology department here, now when he was a postdoc at my lab, did a combination trial in mice showing that when you combine these different checkpoint blockers, it's got at least an additive effect. There was a combination trial initially carried out by Jed Walchuk at Sloan Kettering in metastatic melanoma, showing that combination had a response rate, and here it just shows a 1.5-year survival of over 50%. And I don't have the most recent data, but there's 5-year follow-up now on this combination, and the survival rate is still above 50%. And there's no reason why that won't go on, as ipilimumab by itself did. I don't know yet, obviously, time will tell. But in any event, now we're over half of metastatic melanoma patients, it looks like, can have very durable response to this sort of treatment in a disease which was essentially uniformly fatal when we started this work in 2010. So a lot of excitement there. This is an incomplete slide, but it shows the FDA approvals in different cancer types. Of course, melanoma has been the most, but also PD-1 antibodies and our CTLA-4 antibodies in lung cancer, renal cancer, colon cancer, head and neck, lymphoma, it just goes on, and bladder cancer here. But more interesting, not shown here, is that the FDA has approved the use of PD-1 checkpoint blockade for patients of any kind of histology, and patients that have defects in DNA damage repair and therefore have higher mutational frequencies of neoantigens, and can be expected to respond. The first time the FDA has ever approved a drug for treating cancer based on a genetic defect. But as I said, the tendency is to say, okay, we got 2 checkpoints, we can use them individually, put them together. There's a lot of thought. It's been even written, well, PD-1 has fewer adverse events, so let's use it rather than CTLA-4. But these are that's kind of missing an important aspect of these. And that's that these are very different targets, very different drugs. So I told you, CTLA-4 is hardwired. It doesn't matter what the immune response is, CTLA-4 is going to be involved in terminating that. In keeping your T cells that are starting after priming in the expansion phase and limiting that so that it doesn't kill you. We know that because if you genetically delete CTLA-4, the immune system can't stop, and that's fatal by about 3 weeks to a month of age in CTLA-4 knockout mice. PD-1, as I mentioned, is an induced resistance. It works on fully differentiated T cells. Knockouts there don't have much going on some genetic backgrounds. They can develop lymphoproliferative disorders in some organs, but it really depends on the genetic background. It typically happens only in old animals. Anyway, it seems that CTLA-4 targets the CD28 pathway, PD-1, the antigen receptor, although there is some controversy there, but I think the balance of the data suggests TCR. As I said, CTLA-4 works during priming. PD-1 works on terminally differentiated T cells, and this is really important to understand because of what happens when you inject this into patients. Because of the way they work, CTLA-4 can recruit new clones to a response and expand the repertoire, whereas PD-1 really seems to only expand the existing clones. CTLA-4 responses can often be slow, again, because you're starting at the first stages. PD-1 is reawakening the cells that have already differentiated, so it works very rapidly. So I'll show you later, CTLA-4 primarily affects CD4 cells, although it also has an effect on CD8s, whereas PD-1 has no effect on CTLA-4 on CD4 cells, at least as a monotherapy. Then CTLA-4 can move T cells into solid tumors. So the work that Pam's done in the platform here shows that in prostate cancer and probably kidney cancer as well, at least, CTLA-4 can make a cold tumor hot, whereas PD-1 does not seem to be able to do that. As I mentioned, adverse events for CTLA-4 are frequent, PD-1 relatively infrequent, although they still have the same ones and they can be just as bad. Disease recurrence after CTLA-4 responses are quite rare and are generally limited to focal things that can be dealt with by surgery, where disease recurrences after progression with PD-1 occur about 25% of cases, it looks like, in lung and melanoma. So it's quite a bit different. And finally, not shown here is the dosing schedules are very different. CTLA-4 is typically given not more than 4 times in about 3-week intervals, whereas the PD-1 antibodies, they differ, but they generally go on for quite a long time, as long as 2 years of continuous treatment. So again, lots of differences between these two things. And so what we wanted to do when Spencer joined the lab as a postdoc was take an unbiased, unsupervised way of looking at changes that occur in the T cell microenvironment when you do checkpoint blockade. So we turned to the not so new anymore technique of CyTOF analysis, where you have up to 43 parameters that you can look at simultaneously on cells using time of flight, mass spec. I'm sure everybody's familiar with the mechanisms, so I'm not going through that. But so what we did was treat mice with checkpoint blockers, PD-1 or CTLA-4, and then do the CyTOF analysis. This kind of shows what a tumor looks like when you look at, all the immune cells. All the immune cells, just CD45 positive cells. I'm just going to talk about the T cell part of this today, but it's been shown by Matt Gubin while he was at Bob Schreiber's lab at WashU, and who's now in our immunology department here. There are also really incredible changes in the myeloid department as well. So a lot changes when you do this therapy. Anyway, we had in our panel of antibodies markers for live and dead cells, proliferation, no proliferation. We had barcodes so we could tell, analyze samples simultaneously. And then we had a collection of markers specifically for antibodies to all the major transcription factors that are involved in CD4 T cell differentiation for reasons I'll show you later, and also for cell surface markers of CD8 and CD4 T cells. And that's what I'm going to show you today. But for each individual mice, we kept track of what the tumor size was so that we could not only detect changes, but relate them to whether the tumor shrank or not. So we could determine the cells which were important in the response. So this is the setup. I'm going to show you data today from our analysis of MC38. This is a chemically induced colon carcinoma, high mutational burden, very immunogenic, responds very well to monotherapies with either antibody. But we also did identical experiments on B16BL6, which by itself, which is a tumor with very low mutational burden, very poorly immunogenic. It was thought to be completely non-immunogenic until we showed that at least if we combined a vaccine with checkpoints, we could get responses, but it took a vaccine. We did the same analyses there and got the important cells turned out to be exactly the same in both treatments. So I'm only going to show you the methylcholine MC38. So just a simplified view of what you see is shown here. This is a dot plot showing gating on CD3 epsilon to look just at T cells, dividing them into 4 populations. Here would be FOXP3 positive CD4 cells, FOXP3 negative CD4s, CD8s, and then NKT cells or gamma delta cells. And so looking first at NKT gamma, you can see that that's just a random association of spots. So there's really no action there by any antibody relative to the control. In the Tregs, you can see that the red dots, the CTLA-4 dots, basically disappear. The remaining ones, PD-1 and control dots still look to be pretty homogeneous. This is because CTLA-4, at least the antibody that we use in mice, can deplete Treg cells based on the fact that CTLA-4 Treg cells in TILs express about 2 logs more CTLA-4 than effector cells. And this antibody binds very well to activating FC receptors on macrophages. So it supports T cell killing of Tregs by macrophages coated with antibody. I will just tell you right now that we don't see this in humans. Ipilimumab, the antibody that's used, CD4 antibody that's used, has a defect in its FC receptor, and it does not support ADCC. So it uses only the cell intrinsic pathway of checkpoints. Similarly, tremelimumab, another CTLA-4 antibody in the clinic, has an FC receptor that does not support FC receptor binding either. Anyway, Tony Rebus has looked very closely in melanoma, and in the platform here, we've looked very closely in bladder cancer and kidney cancer. We see no evidence for depletion of Tregs in patients treated with anti-CTLA-4. So that needs to be considered a unique feature of this mouse antibody that we're using. And so we can sort of discount what is associated with that. But anyway, you can see if you look at the CD4 effector cells, the FOXP3 negative, there's a lot of red, very little blue. So that's a lot of effect of those cells by CTLA-4. You look at CD8, it's mixed. You can see patches of red and also patches of green. So there are some subsets of T cells that seem to be affected by both, others just by CD8s. And so to really sort that out, this is a phenogram where these are the different clusters up here, the 15 different clusters that we identified, and all the markers that we used, these phenotypic markers down here. And when we first started looking at this data, I used to tell Spencer, this gives me a headache, how do you deal with this? So we just put it in a simplified version like this so we could look at it. And again, what we found shown here, the Tregs, there are 2 types of Tregs. One has KLRG1, one does not. They're both FOXP3. Both of those are associated with larger tumor size. And you can see that CTLA-4 very depletes a lot of those. In PD-1, the numbers goes down a bit, but we think that's mostly due to dilution effects by the expansion of other cells. In any event, if you look at the CD4 effector cells, CTLA-4 incredibly expands a population of Th1 cells that express TBET and make gamma interferon and TNF alpha. We call these Th1-like rather than Th1 because they express an unusual molecule for T cells called ICOS. I'll come to that in a minute. That was, by the time we did that work, it was already known because of work of Pam Sharma, but anyway, it's an unusual type of T cells. And these cells look like undifferentiated T cells. And in any event, which they're not associated with any changes. So these are the most tumor inhibitory cells in the CD4 population. But if you look at the CD8, CTLA-4 also expands these cells, these CD8 cells. Now these cells are PD-1 sort of low, meaning they've been activated, and CD69. They're TBET, so they're differentiated to be toxic cells, cytotoxic cells. They also have KLRG1, so these are fully differentiated effector CD8 cells. So CTLA-4 expands those, and another population of very not fully differentiated CD8s, and also some other cells that really don't matter much in the immune response are these cells, which actually get in the way of the response. But if you look at PD-1, it doesn't do anything to the CD4 compartment, but uniquely expands these PD-1 high, TIM3 high, LAG3 high CD8s, which have the phenotype of totally exhausted cells. And CTLA-4 does not affect those at all. PD-1 also expands these effector cells, just like CTLA-4 does. And so just to summarize, well, to show you the data first, this just shows that these Th1-like effector cells are associated with smaller tumor size. And this data, by the way, is a compilation either of the control, the anti-CTLA-4, and the PD-1 trials. So it's just telling you those cells, if they're there, regardless of the treatment, correlate with smaller tumor size. This other cluster, which looks like Th0s, don't do anything. In the CD8s, again, you have these PD-1, TIM3, LAG3 high exhausted cells. They also have associated with smaller tumors. And these effector cells, which are Iomes and KLRG1 positive, they also are associated with smaller tumor size. These others don't matter, and these actually seem to get in the way. These look like naive cells that may be just passing through. So to summarize this, so CTLA-4 monotherapy gives you these CD4 ICOS positive effectors and CD8 effectors. PD-1 gives you those same CD8 effectors, increases in them, but also these exhausted cells. Okay, so just to deviate for a moment, what about these ICOS positive cells? Well, what is ICOS? Well, it's another member of the CD28 CTLA-4 family. It's a little further distantly from CD28 CTLA-4. It has its own ligand. It's usually, if you talk to an immunologist about ICOS positive CD4s, they will tell you it's associated with follicular T helper cells, which are found only in to be found in germinal centers in the lymph nodes and spleen, in association with B cells. They're involved in making type 2 cytokines. They're involved in B cell immunoglobulin class, which function would have questionable role in the tumor. And also, it's good, also with a subset of Tregs that make IL-10. So neither one of those would be good. They'd probably be detrimental in the tumor. So this is really a surprise when Pam first showed that in her pre-surgical trial in bladder cancer in 2006. It showed that they were expanded there. Also, it's been since shown with additional studies first in melanoma, in collaboration with Jed Wolchok, and also in kidney cancer. It turns out any cancer that's been associated, that's been treated with NSC4, you find these cells. And again, Pam noticed this first and did a bunch of clinical studies showing that they're a pharmacodynamic marker of NSC4 activity. He also did work showing that if you try to do CTLA-4 therapy in ICOS or ICOS knockout mice, you lose efficacy of the blockade. And later we show that it can't be targeted. And this just shows the study that, mice that have an 85% or so response, wild type mice, to NSC4 therapy. If you do it in ICOS or ICOS ligand mice, you lose more than half in a tumor effect. So this plays a critical role, causal effect of CTLA-4 blockade. So this raises the question of, well, maybe the fact that these are unusual cells, maybe negative co-stimulation has an effect on regulation of T cell differentiation. And this was brought up because when we did this same sort of analysis, they're not with just, if you used NSCD4 and ICOS, you could see some of these cells in wild type mice and lymph nodes are in PBMC of patients not treated. But we could not, with that panel, with all those markers in it, we could not find these cells either in normal human PBMC or in lymph nodes of wild type mice. And so we'd known this for some time, but decided finally to use the Cytol technology again to look at wild type mice with CTLA-4 knockouts or hits. So this again just shows you it's supposed to be rotating. Okay, anyway, what you can see here is again, wild type and knockout and hits are in green and blue. This down here, looking like Australia, are cells that you find in the knockout mice. They're very distant from the others. We cannot, again, not find those in wild type or hits. It's like Australia, it's way far apart. They drink different beer, they sound different. And so, well, what are they? Well, here are these clusters that you don't find, and you can look here, not found in spleen cells, in PBMC, in humans or in normal spleens, in mice. These then are those ICOS positive TH cells. These are exactly the cells you get with the antibody. But we also have these weird looking FOXP3 cells, and these cells that should be at TH2, but have a bunch of other strange markers. And these, which are particularly weird, that have three transcription factors that ought to send them down different pathways, but they've got all three. And they make the cytokines associated with all of them. So these are very mixed up. And this is important. So we have to do a little background immunology 101 here. CD4 is developed, they come out of the thymus, and all they are, what we call a TH0, they really don't have a function associated with them. They have a repertoire and they have CD4, but they're really not committed to any particular function. And what they end up doing functionally depends on the environment they're in when they see antigen and what transcription factor they end up with. For example, cells that end up with TH1, with TBET, end up being TH1s. They make type 1 interferon cytokines, interferon, TNF alpha. Those that end up with GATA3 make type 2 cytokines, IL-4, IL-13, are like TH2s. Sorry, and then those that get GATA3, GATA3 here are TH2s. BCL6 ended up being more like the follicular T helper cells. Those that have ROR gamma T end up being TH17s and make IL-17. So again, the functional characteristics of T cells, CD4 T cells, are determined after they encounter antigen. In other words, what they see during and in immediate steps after priming. And so you could cluster them by the techniques I've shown you, but you could also do an analysis called archetype analysis, where you start with one point and to see whether, you've got this sort of appearance on a curve, on a graph, you don't know whether cells have gone from here to here and these are relevant, or what stages they've gone through. But if you start with one, like a TH0, and you look going further away, instead of looking for cells that are more similar, analyze them for the ones that are a little bit more dissimilar, a little bit less, more dissimilar, more, you can come up with a true pathway lineage analysis, which are called archetypes. And so if you do an archetype analysis, then it looks a lot like the clustering, but what you can see is that these clusters here, 5, 6, and 7, that are as far away from normal as you can get. And if you look at what they are, again, these are those ICOS positive cells that you see when you give the antibody, and then these are two of the other weird phenotypes. 6 are the ones that have the combined, the really combined transcription factors that look like intermediates. So why is this happening? Well, they suggest a number of possibilities. One is maybe the thymus was different. Well, that's not the case. We did a very extensive phenotypic analysis of developing thymus. There's no changes. We even looked at the repertoire, the T cell repertoire, no difference between the CTLA-4 knockouts or the hets or the wild type. Enhanced proliferation of one or activation, no, we could show that those, there's no real, differences between those. So the other possibility is maybe there's dysregulation of phenotypic constraints when they're developing in the absence of the inhibitory molecules. And the easiest way to think of that is that CTLA-4, by limiting co-stimulation, really is limiting the overall effects of signal 1, signal 2, that is T cell activation, to a certain point. And T cells always have some CTLA-4. And so to some level or another, they're always constraining T cell responses. But if you take it away, the T cell signal strength can be, much stronger, because you get, the full TCR signal and unlimited CD28 co-stimulation. And there's some support for this in early work by Kim Bottomley, who showed that stronger TCR signals tend to give you TH1s over TH2s, and weaker signals conversely give you more TH2s than TH1s, for example. So this raises the possibility, again, that what we're doing is that CTLA-4 constrains the phenotypic space that's available to these T cells that are developing after their initial contact with antigen. And when those phenotypic boundaries are removed, you see, for example, the ICOS positive TH1-like cells that you wouldn't see before, plus those other phenotypes. And the other thing that you can do is go back and track individual things when you, along the line of that phenotype, that archetypical analysis, and measure any marker you want as it changes during that pseudo time after the transit from TH0 to the differentiated CD4 type is shown here. An example of that, this goes back to that weird FOXP3 positive cells, but if we just look at that archetype 7, which are these ICOS positive cells that we see with the antibody as well, you can see they start out with high Tbet, they start out with high GATA3, high Tbet, and high BCL2, and with time they lose the other two and end up just expressing high levels of Tbet. And so they go through these intermediate stages and end up out here pretty far from normal as they develop. So anyway, this suggests then not only more nuanced model of CD4 functional development has been suggested by John O'Shea and the late Bill Paul, both NIH, but suggests that this may be responsible, this nuanced pattern of T cell differentiation is what allows CTLA-4 to generate this novel subset of CD4 T cells. Okay, and so it does not change anything really the rate, changes the relative amounts of the CD8s, which are all up here, but doesn't really give you anything that isn't found in the knockouts, as shown in the red and green. So those, the absence of CD4 doesn't have anything to do with CD8s. And similarly, loss of PD1 changes the ratio of some of the phenotype, CD8 phenotypes, but does not give you anything that's not found in the wild type. And so that's because CD8 T cell development is somewhat different. And as I said, PD1 acts only on terminally differentiated effector cells. Okay. So what this tells us again is that I think CTLA-4 is taking advantage of this more nuanced way that T cells, CD4 T cells develop. What it tells you is you've got to really consider when you treat with anti-CTLA-4 or PD1 in the clinic, there are different mechanisms that are involved getting you those cells that are end up having the function. You're not just allowing and generating, expanding a previous existing subset or allowing proliferation of cells which are already there, but you're generating perhaps new ones. At least that's what we propose with CTLA-4. Okay, but what happens when you put these together? Now, how do they interact? And basically what we're saying is, are they much better when you put them together? Because now you've got, instead of just each giving you, a unique subset plus two that are one that are the same, so A plus B equals AB, or is it really different? And so we repeated the same analysis, in this case, just adding a fourth group with the combined therapy. And so then back to these exhausted cells that are expanded by PD1, but not CTLA-4, these PD1 high, LAG3 high, TIM3 high. When you add CTLA-4 back to the mix, those actually go down. So they tend to go down below control levels. They don't go up anymore, they go down. They also don't matter anymore, as I'll show you in a moment. But if you look at the terminally differentiated effector cells, they go up when you combine the two. They go up more than what you see with the monotherapies alone. And so again, to go back and look at the phenotypically exhausted cells, with monotherapy or the controls, if you look at the number of those cells, it definitely correlates with smaller tumor size. But when you add CTLA-4, they're smaller in number and they also don't matter anymore. So it's only the effector cells that matter. And here you see that when you do the combination therapy, that is, you add PD1 to these cells that are expanding, you get further differentiation, you get further proliferation of the Th1 like CD4s. That's because they also express PD1, and so they proliferate more. And so you basically again, decrease the exhausted cells, increase the CD8 effector cells, and increase the CD4 effector cells. So that's just summed up here. So the combination therapy isn't the sum of the two. It's further increases of this one cell, which you get just with CTLA-4, and further increase of these cells, which you get with both monotherapies. And so what happens to those exhausted cells? Where do those effector cells come from? CD8s, well, maybe we're converting them back. And that's a possibility formally, although it seems unlikely because John Wherry and Andrea Schrödinger have both shown that there are epigenetic changes that occur during that transition to those triple positive cells. And so they should be fixed. Now, Didham in the lab has started some experiments where we're going to look for traces of the epigenetic marks on those cells from that arise from either monotherapy or the combination therapy to see if there's any evidence for that. The interesting possibility too is that it's prevented the further exhaustion of those CD8 effectors is actually prevented when you have allowed easy co-stimulated CD28 co-stimulation because CTLA-4 is not around anymore. The cells can not only proliferate more, but undergo repetitive co-stimulation, particularly when you begin to get tertiary lymphoid structures in the tumor, which we know correlates with good responses. And that may prevent actually the CD8s from ever getting exhausted. An interesting idea because it may suggest that that we could think about whether we really need, particularly with combination doses, to give continued exposure of PD-1. And I bring that up now because I forgot to mention earlier the fact that when we treat with PD-1, we can see proliferation of cells with that PD-1 high, LAG3 high, TIM3 high phenotype, that they still look like that, is that the PD-1 antibody is allowing them to proliferate, but it's not changing their differentiation state. So if you take the antibody away, they're going to stop. And so you probably have to keep giving the antibody for a long time with monotherapy, but maybe you don't with the combination. I just don't know, but it's something to think about. And finally, we're beginning to see a difference in some work that Stephen Mock's been doing in the lab. I'm just going to show one experiment there that he's done. And this is not really proper tumor work, but what we did here was just look at responses to a vaccine, in this case, an irradiated tumor cell vaccine, where we supplemented the vaccine with the different antibodies or not, and then re-challenged the mice starting 80 days or so later. And what you can see is the control here. There's really not much memory with naive cells. And if you don't do any antibody, there's not much memory. With CTLA-4 monotherapy in the green, you can see very nice memory response there. But with monotherapy to PD-1, not so much. Again, slightly more with the PD-1 treatment, but in the combination, it's slightly better, but most of that's due to CTLA-4 therapy. We don't know how you can, whether this directs relately or not to the longevity of therapy, the durability of therapy in the clinic, because there's so many other factors such as adverse events and other things that can influence that. But I think biologically it suggests that there is possibly that CTLA-4 plays a bigger role in regulating memory than PD-1 does. In any event, I'd like to just point out that that's the last data slide, but there are a lot more checkpoints out there. This just shows some of them, but Vista is another one that looks like it's important. We know from work, a lot of labs, including the platform here, that after treatment with CTLA-4, you can see Vista, for example, in prostate cancer be induced, which may be some additional target that we have to look at. So a lot more analysis to do. So there are a lot of combinations to be done. Combination of checkpoints themselves, combination with a variety of conventional therapies, and with targeted therapies. As I said, since these kill tumors, and at least CTLA-4 works on at the level of priming at that level, then that ought to enhance the effectiveness, especially of anti-CTLA-4. There are about 3,000 trials going on now. And I would point out that most of these are just looking for clinical signal, which unfortunately I think is a mistake, because if you just reject something because you don't get a good clinical signal without looking at mechanism, you may get a partial effect. They may get you partially there, but you're going to miss it. You're not going to understand why they didn't work if you don't do biopsies and stuff. So this is what the immunotherapy platform here at MD Anderson is looking at. That's the whole reason that we developed it was the notion that Pam had in particular, that we need to learn something from every patient other than just the clinical response. So I'll just close by saying this is what we've been used to in cancer therapy for the last few decades. You can do large randomized phase 3 trials and statistically look and see if you increase the mean survival, and if you moved it over, you call it a victory. We know from CTLA-4 you can do that, but you can also do something better, and that's have a tail on the survival curve, about 20%, which is obviously unacceptable. And what we need to do is get to at least this point, and what we need to do is start thinking of ways to get that higher. We know we can get it to about 50% in melanoma now. With more combinations, our goal should be able to do the right ones to get that as high as we can get it in as many different kinds of cancer as we could get it. Right now we're not doing very well in glioblastoma or pancreatic cancer, and the rest of the cancers are not responding quite as well as melanoma. A lot of work to do, but the good news is we know the basic rules now, and we know we can do it to some degree. So it's just a matter of hard work and and smart work and getting a lot of data and analyzing it. So with that, I'll stop. I hope I haven't gone over time. And I think as George said, we'll be glad to take questions from people who are listening. And thank you very much for your patience.
Thank you very much, Dr. Ellison. This was a fun, wonderful lesson of immunology, simple and very efficient. And in fact, we have a lot of questions, so the attendee were very, very careful. So I will start with the question of a survivor with a 100 complete response to combination therapy, who also say, thanks for your work, MD Anderson, you are saving lives. So thank you very much for your opinion. And the question is, how is the gut microbiome studies going relating to helping the response to checkpoint molecules?
Well, that's a question, and obviously Jen Wargo has a lot of data suggesting it really does. But I must say, at present, we don't know what that is. I mean, there's, something obvious. Maybe there's some kind of antigen mimicry. I doubt that, though. I think it's more likely some indirect effect on the immune responses through toll-like receptor signaling or something like that. It's not just the gut microbiome, which could certainly have an effect, but we're learning now that there actually are bacteria in a lot of tumors themselves, but the mechanisms of it are subject to a lot of investigation, a lot of labs all over the world. We're lucky enough to have a collaboration with Jen doing some correlative studies in mice, different kinds of knockout mice, to take a look at that. It's a very active area of investigation. Hopefully, we'll get answers, begin to get answers soon.
Thank you. Another question. Has it been investigated how much of the anti-CTLA-4 efficacy in mouse models is attributed to its ADCC effect?
Yes, we found with different antibodies it contributes different amounts, but definitely that contributes in mice. Although there is a cell intrinsic pathway that we identified years ago, and that's the best evidence for that is given by some work that we did, and also Arlene Sharp did, where we took T cells that had TCR transgenes, so with known specificity, that would bred onto CTLA-4 knockout mice, and then put those cells, either CTLA-4 sufficient cells or CTLA-4 insufficient cells, into wild type mice. So everything else is the same, and then immunized them. And the CTLA-4 deficient cells still proliferated much longer, much further than the wild type. And so it's hard to say what fraction of it there is, but it definitely contributes. And there are responses, in IPI. I should say that there's an effort being made at Bristol Myers Squibb by Alan Corman to make son of IPI, where they're trying to engineer the FC receptor to make it work, to give it back that particular property. So far, that work's underway. I'm not sure how far it's gone, but TRIMI, another antibody that's there, also has efficacy, and it cannot bind to FC receptors. So that pathway is definitely effective. Unfortunately, right now, that's the only one that we've got in humans, but if we could combine, that would probably make it work better in humans as well. But the bad side was there probably going to be more adverse events with that too.
Thank you. Another question. What are currently the most promising strategies to improve CTLA-4 blockade therapy efficiency in low mutational burden cancers?
Oh, there's a lot. Some are using, suggest chemotherapy, particularly of types that induce mutations themselves. That, of course, is a little bit dangerous. The other is to just increase priming a number of ways by adding TLR agonists. It'll just, get more T cells there, more efficient antigen presentation, and then to kill more tumor cells and induce, more effective priming. We know, for example, that we had a preliminary paper published by work of Sumit Subhuti with the platform, showing that even in prostate cancer, in patients that had only a dozen or so mutations, that we found one patient that had only 2 mutations, and yet with IPI, They weren't detected until IPI was given, but 2 of those mutations at least were recognized by T cells in those patients after therapy. And so, it can be done.
Another question. What do you think about the effect of myeloid cells in the checkpoint response therapy? I'm sorry, I cannot let the name of the people who asked the question, but I will try to put as many questions as possible. So,
Okay. Yeah, I just talked about T cells, but that's a limitation of time. We definitely saw changes in our studies in the myeloid cells. Our panel was set up, even with a small number of myeloid markers, we could see changes. But Matt Gubin, as I mentioned, carried out analogous studies, very much similar, but with a panel that was more focused on the myeloid cells. And there are definitely, with treatment, there are shifts of the different kinds of myeloid cells. You'd have to read his paper to see all the details, but basically what we see, even though these antibodies are targeting, the T cells, there are indirect effects, probably because of cytokines, and maybe other effects too, but you begin to see a shift in certain myeloid cells. It's a lot more complicated than this, but sort of the general, the M2 sort of phenotype, which it has a suppressive phenotype, to a more M1-like phenotype. And again, that's a gross oversimplification, but to a type that does more to help an immune response rather than inhibit it. So definitely big effects.
another question. So we will take a couple more questions. Did you ever profile lymphocytes in the setting of pancreatic cancer? If yes, what do you think about the unresponsiveness of pancreatic cancer to immune checkpoint blockade?
Yeah, that's a wonderful question. That's been done a fair amount. Untreated pancreatic cancer looks pretty cold, but on average, if you look at it, it's not as cold as prostate cancer untreated. There are a few T cells there, but it's got a lot of features. It's got a high myeloid concentration. It's got physical structures, a lot of stroma there, a lot of barriers. And I think the challenges there are some, but I think again, it's an area of active investigation by a lot of people on campus. And I think that in the platform we're beginning to look at it. I think what we've got to do is not just look for clinical signals in combination we do, but do the sort of analyses that I've shown you and say, what happens when you add the antibodies together? What happens if you put in a TLR agonist? What happens if you use something to induce some more mutations? We need more studies, mechanistic studies. But I don't think that it's a hopeless task. I think there's a lot can be done because there are infiltrates there. Just myeloid cells, I think, dominate as it sits with no manipulation.
Thank you. So I would take one more question. I would like to ask, what is the ideal CD8 cancer cell ratio to have an effective killing response in solid tumors?
The ratio of CD8s to what? To
CD8 to cancer cells.
Oh, I don't know. It depends on I guess how effective the CD8s are and their ability to carry out serial killing. Um, um, you know, CD8 cells can kill and then go on and kill, but I suppose different cells may be equipped to kill more than others. So I don't think we could really it may be possible eventually to put a really lower limit on it, but I don't think that we'll ever find a real threshold because there's quantitative and qualitative effects as well that both come into play.
Thank you very much. I think this is a wonderful presentation, and also please go to PBS and check on Jim Ellison Breakthrough documentary. Dr. Ellison, thank you very much for your time, for the presentation, and much more, showing us what means to be, in fact, full of passion for science, and what means to bring as much as possible everything from fundamental science to clinical science and saving lives. And now I want to thanks to everybody who is attending. You guys are wonderful. You are part of our large MD Anderson family for all over the world. We are waiting for you next week, other wonderful presenters, as you can see. And keep in mind, after rainy days come all the time sunny days. What is bad will pass, and we'll come back to what we love more, doing science and taking care of our patients. Thank you to all. See you next Tuesday.